Hey everyone, and welcome. Today we are diving deep into the hidden world of how plants work. We're going to follow the incredible journey of a single cell that gets chosen for a very special destiny. It's a process that is the absolute foundation for how a plant creates a seed. So let's get right into it. So, have you ever really thought about this? We see flowers bloom, and then magically they turn into fruit. But what's the very first step? What happens deep inside that flower to kick the whole thing off? Well, the answer begins with the creation of one single vital cell, the megaspore. So here's our game plan. We'll start inside the ovule, see how one cell gets a special assignment, follow it through a really key division, witness the actual making of the megaspores, and then we'll finish up with a final chromosome count to make sure all the genetics make sense. All right, our story kicks off deep inside a developing ovule. Now, this is the part of the flower that is literally destined to become the seed. And for this whole breakdown, we are going straight to the source material. Everything we're about to cover comes right from these notes, and our job today is to bring this process to life for you. At the very heart of the ovule is a bunch of tissue called the nucellus. The best way to think of this is like a nursery, right? It's this community of cells where our main character is about to emerge. Now, what's so cool about these initial cells is their potential. First, they're undifferentiated, which just means they haven't decided what they want to be when they grow up yet. They're also homogeneous, so they all pretty much look and act the same, kind of like blank slates. And crucially, they're meristematic. It's a bit of a mouthful, but it just means they're like plant stem cells with this amazing ability to divide and become something totally new. And that potential is about to be unlocked, because out of this whole crowd of identical cells, one is about to be singled out for a very, very special purpose. Just beneath the surface layer of the nucellus, a single cell, they call it a hypodermal cell, which just means under the skin, it gets the signal. It's time. It stops being just another face in the crowd and begins to change, to differentiate itself. This chosen one now gets a new name, the archisporial cell, and that name is a huge deal. It basically means it's the ancestor, the starting point for the entire line of cells that's going to lead to the female egg cell. And you can literally see the change happen. This archisporial cell starts to grow, getting way bigger than its neighbors. And its nucleus, the cell's control center, it becomes much more obvious, really prominent. It's basically bulking up for the important job that's coming. Okay, so our special cell is prepped and ready to go. Now it's time for its first big move, a division that's going to set two very different fates in motion. The archisporial cell divides using mitosis. Now, if you remember your biology class, mitosis is basically the cellular copy machine. It makes a perfect, genetically identical copy of itself. And this division gives us two new cells. The first is the primary parietal cell. Think of this one as the construction worker. Its job is to go on and form protective layers. But the second one, that's our new protagonist, the primary sporogenous cell. And this is the one we're going to follow. So while that one cell is off building walls, our main character, the primary sporogenous cell, is destined for a much more dramatic transformation. But here's where things get really interesting. You'd think it would just keep dividing by mitosis, making more and more copies of itself, right? Well, it doesn't. It stops. Its journey isn't about making more of itself. Instead of dividing, it focuses all of its energy on changing its identity, on growing and maturing into its final crucial form. And that final form is the Megaspore mother cell, or MMC for short. This is it. This is the cell that holds all the potential. It is now fully prepared for the most important division of this entire story. And that special division is called meiosis. So unlike mitosis, which was the copy machine, meiosis is a reductional division. Its entire goal is to cut the number of chromosomes in half and create genetically unique cells. This whole dramatic process, from the MMC undergoing meiosis to producing the final product, has its own scientific name, Megasporogenesis, literally the genesis or the beginning of the megaspore. And the result? From that one megaspore mother cell, we get a little cluster of 4 brand new cells. These are the megaspores. And the most critical thing to know about them is that they are all haploid, meaning each one has only half the number of chromosomes that their mother cell started with. That whole chromosome cutting business is so important. Let's just do a quick final count to make sure the genetics of this whole thing are crystal clear. So check this out. The cells we started with, the nucellus, were diploid, or 2N. They had 2 full sets of chromosomes. The megaspore mother cell was also diploid. But then, bam! After meiosis, the megaspores that are created are haploid, or N, with just 1 set. And the future female gametocoit that grows from one of them will also be haploid. This is the entire point of the process. So we've followed this amazing journey from 1 diploid cell all the way to 4 haploid cells. It's the perfect outcome of meiosis. But, in the ruthless economy of nature, not all of them are actually destined to survive. And that brings us to our final cliffhanger question. This whole intricate process creates 4 potential heirs to the throne, but only one will actually become the future female gametophyte and carry on the lineage. So, what happens to the other 3? Well, that is a story for our next explainer. Thanks for watching!